Welcome to your online lecture for the course Anatomy and Physiology. In today's lecture, we're covering the lower digestive tract, which is a follow-up lecture to the upper digestive tract that we discussed during our last lecture. I had mentioned that the upper digestive tract really focused on our mouth and our pharynx through to our esophagus and down into our stomach. And that's where we ended off. And then the beginning of the small intestine through to the large intestine and so on and so forth includes our lower digestive tract. And within this lecture, we're also including some accessory organs, so organs that contribute to digestion. We spoke about salivary glands, for instance, already during our upper digestive tract lecture. But this one will cover organs such as the liver, for instance, that contribute to or assist with digestion without actually coming into contact with the food that we consume. A reminder that the food from our stomach is going to leave our stomach, so leave the pylorus region of the stomach through the pyloric sphincter, and then it will enter the upper part of the small intestine called the duodenum. Now, the size of the small intestine in terms of its diameter is 2.5 centimeters and about 6 meters in length. So really not very, it might sound big, but not very big when you consider what its potential is. There are three distinct regions: the duodenum, the jejunum, and the ileum. The duodenum, which is only 25 centimeters long, it coils in such a way that it forms a C shape. And it's our uppermost, so the food or chyme moving from our stomach into the small intestine will first reach the duodenum. Then we have the jejunum, which continues then to the ileum, which then will join into the large intestine, as you'll see later. This here is a radiographic image of the small intestine of a human, and you can see an actual image here, likely using some form of laparoscopic, some sort of laparoscopic instrument. Now, even though the small intestine is only 6 meters in length, it has a surface area of 250 meters squared, which for trying to visualize this would be the size of a tennis court. Now, how is this possible given that it's only 6 meters long? Well, we'll talk over our next few slides about some of the features that contribute to this massive increase in surface area relative to its original length. Here you can see a section of the ileum wall. Remember that the ileum is that third portion of the small intestine. And what I'd like you to make note of, first of all, this is the lumen, so this is where food is actually passing through. Remember that that's still part of our external environment. And these finger-like projections here are called villi, and they extend into the lumen of our gastrointestinal tract. And they contribute very much so to this massive increase in surface area. So if you were to think about all of these present within our small intestine, and how if we were to extend it all out and not have these folds, how large our small intestine would actually be. I'd also like to point out the presence of goblet cells, which produce mucus, which is important. And also at the base of each villi, in between each villi, we have intestinal crypts. And we'll speak about their role shortly. I should mention that the main function of our small intestine is where nutrients are going to be absorbed, and so our small intestine is designed in such a fashion that we can maximize or optimize absorption. It isn't just the villi that we talked about on the last slide that contribute to the surface area, but also what are called microvilli. You can see this term here. And if you look up real close, this is where we can see the microvilli, and this contributes to a massive increase in the surface area. And it allows you to see that the ability of the small intestine is really much larger than it appears to be. Each villi is covered by about 1,700 microvilli. You can see a villus here. The spaces in between those finger-like projections, so this region right here, are the intestinal crypts. So we'll look on the last slide as a comparable. The intestinal crypts, the spaces in between each of these finger-like projections. And at the base of the crypts are these epithelial stem cells. And what they will do is continue to divide and produce more and more cells. And these cells will migrate up towards the top of the villi and replace old cells. So we have this cell shedding that occurs, and it's very similar to what happens when we shed skin. Old cells are shed as dead skin, and new cells are replaced, and it allows for a continuous, healthy, fresh, optimally functioning layer of cells. Now, as we move from the jejunum, so the second part of the small intestine, to the ileum, then to part of the, and we'll talk about this shortly, the large intestine, you can see that the projections into the lumen change, those villi projections, and we don't have them. And the reason for this is because absorption within our small intestine, and the cecum is that first part of our large intestine, rather, the absorption within our large intestine is not the critical role. We still have a thick mucosal layer, but we don't have the same projections. So these are actual images of the small intestine, and getting into the large intestine and the colon, and then the histological samples beneath. So again, the key point to take is that we don't have these villi present once we enter the large intestine, because its main role is not that of absorption, where we need to maximize surface area. The large intestine is significantly shorter, anywhere from 1.5 to 1.8 meters in length, and only about 6 centimeters in diameter. So it is larger in diameter, but not in length. And to orient yourself here, the small intestine, for the most part, has been removed from this image, but you can see the end part of the small intestine, so part of the ileum here, where it joins into the large intestine. And once we're in the large intestine, this is the region of the cecum, the first part of the first part of the large intestine. And then as we ascend, it's called the ascending colon. Along this region here, we have the transverse colon, and that's because of the orientation of the gastrointestinal tube. Then we have the descending colon. Here we have the sigmoid colon. It's all listed here. The sigmoid colon, and it gets its name because of its S shape. And then we have the rectum, and then finally the anus, where food is, or feces really, is evacuated. The intestinal mucosal glands are going to produce a lubricating mucus that is going to coat the feces as it's formed to allow for nice fluid movement. Otherwise, there would be a significant amount of friction that is present. And remember, we spoke about goblet cells and secreting the mucus. I'd also like to mention the presence of rugae. I mentioned this term in our upper digestive class. You can see the presence of rugae here with the folds or ridges. And we'll talk about it in an upcoming slide, but here is what you all would know as being the appendix. It's called the vermiform appendix. It gets its name from its shape of a worm. We'll talk about its role shortly. So again, food has moved from in the mouth to the pharynx. Then it's moved down through the esophagus into the stomach. Once it moves from the stomach into the intestinal tract, it's moving from the duodenum into the jejunum, into the ileum, and at the very end now of the ileum, food is going to enter into the large intestine and make its way through. Now, the rectum is the final last, say, 7 or 8 inches of our intestinal tube, and you can see the presence of these rectal folds here, or sometimes called rectal valves. And the final terminal inch is called the anal canal, and this is where the final leads to the final opening that's called the anus, where feces is evacuated. And there are 2 types of sphincters for you to be aware of. We have the internal and the external sphincters. The internal sphincters are not under voluntary control. The external sphincter is under voluntary control, and it's the external sphincter that allows us to control defecation. And in some conditions where there's loss of this voluntary control of the external sphincters, it could be some sort of medical emergency. Onto the vermiform appendix. It's considered an accessory organ, a small accessory organ for sure, meaning that it is helpful to some extent for digestion. And it branches off the cecum, as we saw earlier. And you can see it really tiny right here. And I'll show you better, go back to this better image here. It's really the first of the accessory organs that's involved in digestion, and it's really the difference between this accessory organs and other ones such as the liver is this is an extension right off our digestive tract itself. Now, its precise function is uncertain, or maybe even you could say argued over by researchers, but it's considered to be and generally accepted to be some sort of reservoir for beneficial gut bacteria to some extent. And beneficial gut bacteria will either directly or indirectly benefit our body and also benefit themselves. So the presence of this bacteria benefit themselves. Maintaining what we would call a normal intestinal microbiome helps prevent pathogenic bacteria from becoming established. So when we have the introduction of pathogenic material into our intestinal tract, normal, good, healthy bacteria will help kill that off. Now, when the normal microbiome of the gut is disrupted by maybe an infection or maybe by antibiotics, we do have bacteria that's hidden away in the appendix. And when, say, we have a loss, if somebody takes antibiotics, which enters through the stomach and will pass through our digestive tract, and systemically throughout our body, these antibiotics kill bad bacteria, but they also kill good bacteria. And that good bacteria is vital in many ways for proper digestion, but also for fighting off further infection or pathogens. And so when that is lost or reduced, the appendix is thought to be a storage site of this good bacteria. And so when it's needed, they can migrate into the colon, these live bacteria, and at least begin the process of restoring the normal ecological balance. But this process takes time. The large intestine is an ecosystem for microbes as well, which you can see in the diagram. What you can see here is that there are the microbial load per milliliter, and it's present all throughout our intestinal tract. But what you can see is the relative increase in microbial load as we move from the stomach to the large intestine. You can also see that depending on where we are in the intestinal tract or through our digestive tract, there are not only a different quantity of bacteria, but different types of this good bacteria present. Then if we look specifically at the large intestine, just by viewing this with your own eyes, you would agree that it is a very diverse ecosystem. In fact, it's the most diverse ecosystem within our body in terms of microbial load per milliliter and the various different types of good bacteria that we have present. And in the large intestine, the average person's large intestine, there are at least 30 trillion bacteria present, and we have about 30 trillion cells in our body. So that really demonstrates how much we have within our large intestine alone. Onto the peritoneum. The peritoneum are considered large continuous sheets of serous membranes. Remember, we talked about the serous layer when we spoke about the different layers of our digestive tract or of our GI tract. It included that serous membrane, but we will collectively call that then the peritoneum. And the entire abdominal cavity would be then collectively referred to as the peritoneal cavity. Many organs within our digestive tract are covered by what's called visceral peritoneum. Remember, the term visceral refers to organ. So this is a peritoneum or a serous membrane that's actually covering a particular organ, which you can see identified here. We also have a parietal peritoneum, which lines our abdominal cavity. And this is all continuous in some way or another with each other. The mesentery, speaking sort of generally, is a fold of tissue that attaches organs to a body wall. So it attaches it to the body wall, but it also allows for its free movement throughout each coil of our intestinal tract. In this image here, you can see a normal liver that's being prepared for organ transplantation. The liver is considered an accessory organ of our digestive system. In this image here, it is clear that it's a healthy liver that's smooth, it's pink, it's plump, it looks healthy, especially when you compare it to a liver such as this that is damaged from, for example, excessive alcohol consumption over a long period of time can lead to cirrhosis of the liver. The liver is the largest gland in our body, and it weighs about 1.5 kilograms. In this left diagram here, you can see both left, remember to orient yourself, and right lobes, which are divided by what's called a falciform ligament that divides both of the lobes. If we were to look at the underneath surface, as in this image here, the left lobe this is the underneath surface, but let's look back here for just a moment so that you can see that the left lobe is only about, say, well, probably in and around a sixth or so of the liver, so it's much smaller than is the right lobe. Now going back to this image here, the right lobe can be further divided into 3 lobes, and the lobes that you have to be aware of are the right lobe proper, the caudate lobe, and the quadrate lobe. So these are further divisions of the right lobe, which can be more easily seen on the underneath, much more easily seen on the underneath of the liver. The liver also contains what are called hepatic lobules, which we'll speak about in the next slide, and these are considered to be discrete anatomical units. They're considered to be the basic functional unit of the liver, and that's described a lot more on our next slide. The hepatic lobules, when you hear the term hepatic, it's referring to the liver. The hepatic lobules are arranged in a hexagonal shape. You can see it in both images here. So this drawn image, you can see the hexagonal shape, but if you look at this histological image, if you can kind of squint a bit, you can see the hexagonal shape of the hepatic lobules. Remember, these are the basic anatomical unit of the liver, and it's really overall a quite complex arrangement. The liver gets its own blood supply directly from the heart, and that's from the hepatic artery. So just like other organs, the liver itself needs oxygen and nutrients to survive, so the hepatic artery is going to provide that for its survival. The liver receives blood from the small intestine, so this should be a review for you and important to know for your final exam as well, is that the liver receives blood from the small intestine. Okay, when you take in food, this nutrient-rich blood from our GI tract doesn't go straight to the heart to be sent out to the rest of the body. Instead, it first goes through the liver. This is done through the hepatic portal vein. That should be a review for you. And you can see the hepatic portal vein here. Blood arriving at the liver from the small intestine needs to be detoxified and purified before it makes its way back to the heart. Now, more specifically, the nutrient-rich blood from the small intestine that comes into the liver through this hepatic portal vein is going to drain into what are called sinusoids, which is labeled here. So it's going to drain into these sinusoids. And throughout this process, it's going to pass by hepatocytes. Hepatocytes are liver cells. Hepato referring to liver, cytes referring to cells. And this is where the blood is going to be cleaned, so to speak. And this clean, or at least cleaner blood, is going to travel throughout the liver until it reaches the central intralobular vein, which is right here. Intra, because it's within, okay, inside, in between the lobes, intralobular vein, where this blood is going to drain, be emptied into our hepatic vein, which you can see more clearly up here, which is where it's going to be then returned to the heart. So make sure you're aware of the differences between the hepatic portal vein, the hepatic artery, as well as the hepatic vein, because they do have much different roles. Let's speak just briefly now about the bile ducts, which is seen in green on here. So toxic substances, we inevitably take in a fair amount of toxic substances. They will leave the blood and empty into these interlobular bile ducts. Okay, so they're found in between the lobules, the interlobular bile ducts. And we'll speak about some role of bile on our next upcoming slide. So in summary then, the liver lobules are involved in nutrient metabolism, involved in storage, And I'll give you an example of storage would be glycogen. So our liver stores glucose as glycogen so that when we need to increase our glucose levels in our blood, it can be broken down. It's involved in detoxification, which was the main thing that we spoke about on this slide when we discussed the liver lobules. And it's also involved in bile secretion, which will be discussed next. This slide is referring to the gallbladder, so you can see the gallbladder here. And this is where bile is stored. Bile is produced by the liver and stored in the gallbladder. A few important structures to point out here. Each bile duct from the liver are going to come together and form the common hepatic duct. Right here, common hepatic duct. So they come together, form a common hepatic duct. Now, when bile is produced by the liver, it's going to migrate to the gallbladder, where it is stored until it's needed during digestion, and it's involved in assisting with the digestion of fat. Once this common hepatic duct passes the level of the cystic duct here, it changes its name to being called the common bile duct. And at this point, it travels, this common bile duct continues down through the pancreas, passing through and now merging with the pancreatic duct, forming a common duct that will pass through and into the upper duodenum to contribute to digestion. And a couple other structures you need to be aware of. This structure here called the major duodenal papillae, which has muscles that will either contract or relax to limit the passage of bile, as well as any pancreatic juices, into the duodenum. The gallbladder is separate from the liver, but it's somewhat embedded against it, as you can see in this image here. And it can hold anywhere from 30 to 50 milliliters of bile at a time. It's not just responsible for storing the bile, but it can also concentrate the bile. So the gallbladder can absorb certain amounts of water to make the bile more concentrated, and it will also eject the bile when needed, and that's where it's ejected through here into the duodenum. And when it's needed is when we're bringing fats into our stomach, puts chyme into our small intestine, and in the presence of fat, bile is needed. Sometimes the gallbladder needs to be removed for conditions such as gallstones. So this is a case of cholesterol-rich gallstones, and you can see the excessive amounts and size, and this is something that needs to be removed relatively quickly. As you could imagine, if you have stones in your gallbladder and they start to migrate through here, not only will it cause excessive and significant amount of pain, but it will also cause blockages of bile. So the bile that's stored here can no longer pass through, and it can even promote the further formation of stones. This is more common in the elderly, especially the more excessive cases, as well as those that are obese. And on our last slide here, which is, we're discussing the last accessory organ of the GI tract as it pertains to this course, I want you to recall that the pancreatic duct, right here, also visualized here, merges with the common bile duct. And contents then of what's produced in the pancreas can empty directly into the pancreatic duct and enter into, so this is considered the pancreatic duct here, and can empty into the small intestine through the major duodenal papillae. So when we need certain substances, it can be released this way. There's also a minor duodenal papillae where the accessory pancreatic duct, so this is an accessory pancreatic duct, enters into the small intestine as well. The pancreas is a bit larger than the gallbladder at about 12 to 15 centimeters long. The pancreas is an interesting gland because it has both exocrine and endocrine functions, so it makes it a very unique organ. Digestive enzymes are produced within the exocrine acinar cells that are involved in digestion. And what's happening is if they're produced within the pancreas, then these digestive enzymes, as part of from our exocrine acinar cells are going to enter in the digestive tract. And think of EX for exiting. What's really happening is these digestive juices are going to exit the pancreatic duct, and they're going to end up as being really part of our external environment, because we know that the GI tubing is part of our external environment and not part of our internal environment. And this makes up the majority of our pancreas in terms of function, or the production of gastric juices from these exocrine acinar cells, which you can see here. Now, in addition, or a small minority of the pancreas in terms of its function are pancreatic islets. So pancreatic islets found within here, as you can see, pancreatic islet. These have endocrine function. Remember, when you think endocrine, I want you to think of hormones. And there are two types of cells you need to be aware of, the alpha cells and the beta cells that make up the pancreatic islets. The beta cells here are responsible for secreting insulin. So when you've consumed a meal, your body absorbs the glucose from that meal, and it, in varying amounts, and it enters your blood. You don't want that glucose to stay in your blood for too long. And what insulin will do is it will help with the uptake of glucose into our body cells, where we use that glucose to produce energy. We also have alpha cells that produce glucagon. Glucagon is a hormone that promotes really the opposite to what insulin does. When your blood sugar levels are low, so in between meals, in order to have sustained energy and sustained glucose in your blood, traveling to tissues, glucagon will promote the breakdown of glycogen. I told you that glucose is stored in the liver as glycogen, and when glycogen breaks up, glucose will enter into our bloodstream and keep us in homeostatic balance. That's the end of today's lecture. Thank you for listening.